All right. Good afternoon, everyone. It is 1 p.m. on the east coast of the U.S., so it is Eastern time. My name is Riccardi, and I'm happy to welcome our in-person and online audience to today's talk. Before hearing from today's speaker, I want to alert you to our talk in 2 weeks' time when Dr. Shi Young Sun from UAH University will present her work on a cross-linguistic and multicultural framework for aphasia assessments. Today, however, it gives me great pleasure to welcome Dr. Riccardi, postdoctoral fellow here at the University of South Carolina and a member of our C-STAR team. Dr. Riccardi completed his PhD in experimental psychology, studying the neurobiology of language using diverse methods, including brain stimulation, functional MRI, and Leishman syndrome mapping. His work focuses on the determinants and consequences of brain health decline across the lifespan in both normative and stroke survivors, normative populations and stroke survivors. Today, he'll share some of his work on deep learning-based brain age as a biomarker for post-stroke language recovery. During his talk, please feel free to write your questions in the chat box for him to address at the end of his talk. And with that, I'll give the floor over to you, Nick. Thanks.
All right, thanks everybody for being here. So today I'm going to be talking about a relatively new method called brain age and how we can apply it to post-stroke language recovery, specifically for prognosis, and also explaining additional variants beyond the traditional factors that we've been using to try to explain aphasia severity. So first, I'm going to talk a little bit about what brain age actually is, so we're all familiar and introduced to that. We're going to talk about some key findings that have used brain age in stroke-free populations. And then I'm going to talk about the gap that we're trying to address here, which is how can we apply brain aging to post-stroke aphasia. Okay. And so as part of that, I'm going to talk about some of the actual methods that we use to apply these brain age models to brains with stroke lesions. And I'm also going to talk about how we are using brain age to explain variance in baseline aphasia severity and then also longitudinal treatment response. So the idea behind brain age is it's part of a whole family of new methods called biological clocks. And the premise of this is that the brain may actually age faster than the rest of the body due to lifestyle or genetic factors. And we want to be able to predict the biological age of a brain from structural MRI using machine learning. So that's the entire idea behind these brain age algorithms. And then we want to compare the predicted age, so how old does that brain actually look, to the participant's chronological age, and from that we get a brain age gap. So is your brain older or younger than it should be? And this is what that looks like in practice. So we can see here in the top panel is somebody who is 50 years old, and their brain age is 67. So their brain is aging at an accelerated rate. And then down here we have somebody who is 65, and their brain age is 52, right? So this person has a relatively younger looking brain. And this matters because it lets us capture deviations from normative aging and use it as a proxy for brain health and relating that to behavior and lifestyle factors. So here's a typical brain age pipeline, and a caveat here being that there are a lot of brain age models out there that all have their own unique quirks. This is just an example of one of the models. So we can see here in panel A, it's 3 different people. They're all aged 80. We have 2 healthy people and then somebody with dementia. Okay, so we take their brain scans and we put it into a machine learning pipeline. And again, this will vary depending on the brain age model that you're using, with the goal of predicting each person's age based off of their brain scan. Okay, so At the end of that, we get a different score, so the brain age gap, indicating that the person with dementia has a brain that looks like an 88-year-old's, whereas this healthy person has a relatively younger looking brain, right? And then we can do different statistical analyses with these older appearing brains versus younger appearing brains in regards to disease and lifestyle. And all of this, all of the first steps for brain age have been done using a global brain age score. So one number for the entire brain. And the pro of this is that it's a very easy to interpret metric of brain health. You can see the immediate clinical utility of this. So like if you get a brain scan and you want to know how's my brain doing, somebody can say, well, your brain looks like a 50-year-old's, you're 40, maybe you need to drink and smoke less, right? So that's kind of the draw to this type of method. And another thing is that it is not just a measure of atrophy. So things like gray matter volume are what we typically use to assess the structure of the brain. But brain age, because of the different machine learning methods that they use to generate these scores, are actually using multivariate properties of the brain structure itself. So it's not just gray matter volume, for example. It's a completely new metric. And this is shown here in this bottom panel, where the relationship between brain age and gray matter volume or atrophy is they are related, but they are not the same. So some key findings, and I'm going to highlight the work of James Cole here, who is one of the pioneers of the brain aging field. I'm going to focus just on a single paper of his, but he has many out there, and a lot of them are very, very good. So we want to look at how does brain aging relate to health and behavior, basically establishing that it is a meaningful metric. So basically what the premise of this is just taking the estimated brain age or the brain age gap and relating it to, in this case, cardiovascular risk factors, right? So in here, they found that blood pressure was related to accelerated brain aging. So the relationship between cardiovascular risk factors and brain health is then established with this metric. And you can also look at it in relation to lifestyle factors. So smoking and alcohol are both related to accelerated brain aging. And you can also relate brain aging then to cognition. So here in this paper, they found a relationship between accelerated brain aging and worse cognitive performance in a variety of tasks. And then I want to highlight some other findings from members of our group, Sam and Natalie, who published a lot of papers using brain aging in, again, mostly stroke-free populations. So relationships between brain aging and hearing loss, brain aging and socioeconomic status, language fluency, and also relating different properties of the brain to accelerated brain aging, right? So what is accelerated brain aging actually measuring within the brain? And so all of this put together, these are just some of the manuscripts that have been published, but it's all pointing towards the idea that brain age is a meaningful and easily interpretable metric of brain health. And we can see that the publications per year using this metric are rising in the fields of normative aging and also in dementia. But this method does have drawbacks, right? So we all know that using a single number to describe a brain is overly simplistic. The brain is comprised of a lot of regions, a lot of networks that all contribute to their own cognitive processes, right? So using one number is probably not the best way to go about things. When we use one number, we lose specificity, we lose the ability to look at specific brain regions that we know are important for different things, and it could obscure disease-relevant patterns actually within the brain. So that's where I want to introduce the idea of region-specific brain aging. So instead of just generating one number for the brain, we're actually going to generate a bunch of numbers for all of the brain regions in a given parcellation. And for the rest of this talk, when I'm talking about region-specific brain aging, I'm specifically talking about this manuscript and using the pipeline that these authors developed, which is housed within VolBrain. And what it is basically is it's a convolutional neural network that takes in an anatomical scan, so just a routine T1, for example, and trained on very, very large amounts of data. So I think they trained it on 40,000 brain scans. This convolutional neural network learns features from that image directly from the images that are most predictive of chronological age. And the output of that is then within the brain segmentation, we can get regional brain age estimates. So now we're not talking about one number anymore. We're talking about the entire brain and patterns of brain aging. And that's what this looks like once we have these maps. So here we can see these are 3 example participants. And we can see that some people have different patterns of brain aging across their cortex, right? So subject 1 has accelerated aging in the ventral visual stream, for example. So within individuals, there are different patterns of brain health that we can observe. And so the pro of this is that it brings back anatomical specificity. So we can look at individual brain regions, which is very important for what we do with neuroimaging. And this is a very new measure. So this pipeline, this manuscript just came out last year. So not a whole lot has been done using this region-specific approach. And so I'm going to talk about some work now that me and my colleagues have been doing to look at, can we use individual patterns of brain aging? And does that give us a better idea of what's going on than some of these more global whole brain measures? So this regional approach gives us a lot of new opportunities. So like I said, we can characterize region-specific trajectories of brain health. We can identify commonly expressed patterns across groups of individuals. We can relate specific spatial patterns to what is already known about the brain. So this gives us mechanistic insights of maybe what could be driving or what features of the cortex are related to accelerated brain aging. And then finally, and probably most importantly, we can see if different people who have different patterns of brain aging, does that relate to cognitive decline, or in the case of aphasia, worse language outcomes, for example. So now I'm going to talk about kind of a preliminary, well, it's not preliminary, but validating this regional approach using what is already known about the brain. So using neurobiological properties. So what am I talking about when I'm saying neurobiological properties? So the overall concept is that there are different spatial gradients that organize brain structure and function. And so these can reflect a lot of different things. So cortical thickness, blood flow, for example, metabolism. And for the work that I'm about to show, we used it's called the sensory motor to association axis framework. And this is a framework that was built over decades of cross-modal evidence. So this includes in vivo neuroimaging, it includes post-mortem techniques, and it also includes cross-species comparisons. So if you're, for example, comparing human brains to non-human primate brains and seeing which areas expand as evolution has progressed. And the importance of this is that it lets us capture principles of brain organization. These things have been well validated over the course of decades, right? So these are properties of the brain that we know. And the idea is to then see if brain aging relates to these properties. And here's an example of what this looks like in practice. So this is the map of the sensory motor to association axis. So areas in deeper purple are association cortices. So these are areas that are involved in higher cognitive functions. They develop later across the lifespan. So this is the thing that we all hear about, the frontal lobe being not fully developing until we're 25, right? And then the areas of life are prototypical sensory motor cortices. So these are the areas that develop first. They're evolutionarily the oldest. So we can see here the occipital lobes, which are central to vision, and sensory motor cortices. These are the most basic human processes. So they're low on this hierarchy. And within that, we actually have many other maps that map different other properties of the cortex. So these are just some of them, but we have maps of cerebral blood flow, so areas that have high perfusion. We have maps of gene expression, so the brain areas that are most likely to express genes that are related to cognition and mental health, cortical thickness, et cetera. So we have all of these neurobiological maps that we can use. So for this study, we had 335 participants aged 20 to 86, so a very nice range of the adult lifespan. And for every single person, we generated this region-specific map of brain aging. And we wanted to see, are there commonly expressed patterns of brain aging across this cohort? And to do this, we used two separate data sets because we wanted to see if we identified patterns in one data set that they replicated in a completely independent data set. And to do this, we used exploratory factor analysis on the brain age gap correlation matrices. So in theory, this should identify shared patterns of accelerated or decelerated brain aging across regions, and the output should be different patterns of brain aging that are commonly expressed in this cohort. So what we found was that 6 different patterns of brain aging emerged, and these patterns replicated across 2 independent cohorts, so they seem to be replicable. This is not in the final published version. We replicated it in 2 data sets, but since then, I've also replicated it in a third. So these seem to be stable regardless of who is collecting the data or the cohort that's being used. And we can see that these patterns also look interpretable and meaningful. So for example, we have a frontotemporal pattern that emerged as the primary pattern of brain aging. So areas in red are areas where people had accelerated brain aging in those regions, and then blue are areas where it's decelerated aging in this pattern. And we have a pattern of accelerated dorsal stream aging, ventral stream, et cetera, right? So these patterns look sensible. But then the question becomes, are these patterns neurobiologically meaningful? So do these 6 patterns that we extracted reflect what we already know about the brain? So those neurobiological hierarchies that I was talking about before. And so for this, what we used was parcel-based permutation spin testing to compare the different neurobiological hierarchies from the sensory motor to association axis to the different brain age patterns that we extracted. And we want to see if those patterns map onto the properties of the cortex. And long story short, what we found was that the specific brain age patterns do align with different neurobiological hierarchies. So I'll show you what that looks like here. So for right now, just focus on factor 1, which is our primary pattern of brain aging. And we can see here a heat map of the relationship between this pattern and different neurobiological hierarchies. So this dominant pattern of brain aging was significantly aligned with the prototypical sensory motor to association axis, a map of gene expression, external parameterization, and anatomical hierarchy. So that suggests that this dominant pattern of brain aging does reflect properties of the brain that we that are sensible and that we know about. So the correlations down here in black boxes, Those survived FDR correction. So, this is correcting for multiple comparisons and using gold standard statistics for comparing multiple cortical maps to each other. So then the natural next question is, are these patterns that we observed behaviorally meaningful? Do they relate to cognition or other behaviors that we can measure? So I'm not going to get into the super in-depth statistics of what we did, but basically for each participant, when we run this factor analysis, we are able to generate scores that represent how much an individual person's brain age pattern maps onto each of those 6 group level patterns that emerge. So for example, this participant in this panel has a high score in factor 1, meaning that they have accelerated brain aging in the medial frontal areas that were identified in pattern 1, right? And then we can see here, so this person has accelerated aging in the ventral stream, and that relates to the pattern that we identified that was pattern number 3, which was accelerated ventral visual stream aging, right? So for each of these people, we get scores that represent how well their patterns align with how well an individual brain's pattern aligns with the group level patterns. And another important thing that I want to highlight here is that if we were to use a global brain age approach, so just using one score for the entire brain, we would be missing out on a lot of this variability that we are now able to capture. So you can see here, in this participant, their global brain age is actually decelerated. So they have an overall healthy brain according to this model. But even though they have that at the global level, they do have areas of accelerated aging, right? Okay. So basically, we took the participant level factor scores, used them in a linear regression along with like age, sex, and education, so typical demographic variables, and we first related them to cognition, and we also related these patterns to vision, hearing, balance, and gait, so cognition and sensory motor tasks. And what we found was that these patterns were related to these different behaviors in a way that was biologically meaningful. So I want to highlight that, for example, worse visual acuity, so worse vision, was related to accelerated aging in the ventral visual stream, which is it's a very clean and biologically reasonable result that kind of highlights that this method is capturing meaningful variance in people's brains. And then I want to highlight that if we use just the global brain age gap instead of these pattern scores, the global brain age gap was not significantly associated with any of these variables that we measured here in this cohort. So we would have missed these associations if we had only looked at global brain age instead of this pattern-based approach. So the take home is that we took a novel method and we kind of tried to validate it. We found that we were able to identify robust patterns that replicated in independent data sets. These patterns seem to be neurobiologically grounded. They aligned with different properties of the cortex that we know to be governing properties of the cortex. And they were also meaningful. So they related to behavior. These relationships were modest, but we were also using healthy individuals. So these are not people with Alzheimer's. They're not people, with neurological disorders. These are healthy, normative adults. And we were still able to find relationships between brain aging and these cognitive and sensory motor behaviors. We recently published this in Communications Biology. And then briefly, before I move on to post-stroke aphasia, I want to highlight that we're also finding relationships between peripheral biomarkers of health and brain aging patterns and regional brain aging. So, for example, we can look at properties of the DNA. So in this case, we used another biological clock called DNA age. And we found that people with older looking DNA also had older looking brains, but only in some regions, right? And kind of these frontotemporal areas that are important for cognition. And then just briefly, the relationship between DNA aging and brain age also became stronger as we moved higher up into the association cortices. So again, using the sensory motor to association axis. And we published this year in Neurobiology of Aging. Okay, so that was a background of what we've been doing with regional brain aging. And now I want to highlight an important gap. So that is using this method in aphasia. So we can see here that brain age, when used with dementia or cognition, this was just a PubMed search. We're approaching 600 publications per year that are, increasing over the years. But this method has very rarely been applied to look at aphasia. So this is a very addressable gap, and I think it could be a very important gap in the field of stroke recovery more broadly, even moving beyond aphasia. So what is the reasoning? Why do we think that brain aging should be a useful measure to use in post-stroke aphasia? Well, we would expect that non-lesioned areas may show accelerated brain aging, perhaps due to the stroke, so the damage that actually happened, or it could be due to pre-existing factors that we know are nearly ubiquitous in stroke survivors, so like cardiovascular risk factors. And the reasoning also being that accelerated aging could possibly explain variance in language outcomes or in recovery potential that we are currently not measuring. And again, so this approach will provide us with anatomical specificity instead of using just a global measure of brain health. And something I really want to highlight here is that it's something that I think is really important. And that's that this only requires structural MRI, so routine T1 or T2. So there's no tasks, there's no specialized acquisition, there's no, research center specific analysis pipeline that needs to be used in order to replicate these results. It's just a T1 fed into a pre-trained deep learning model, which I think is a very scalable approach, and it could be a very clinically applicable approach because of its simplicity. So I'm going to talk about this study that is currently submitted, so it's not published yet. I want to highlight my an undergraduate that we are mentoring, Ainsley Martin, who is co-first author on this project, and she's been doing great work on this. And so our key ideas were, we wanted to identify brain aging patterns, so similar to what we did in the normative sample. And we wanted to do that in our stroke survivor cohort. And we focused on the right hemisphere because the left hemisphere has the stroke lesion, right? And I don't want to get too into the details, but basically when a stroke lesion is present, it makes the brain age estimations unreliable in the current models that we have. So that's why we want to focus on the intact right hemisphere. And we want to see if these patterns relate to aphasia severity, so as measured through the Western Aphasia Battery Aphasia Quotient and the Philadelphia Naming Task. And we want to see if it explains variance beyond what can be explained with the lesion characteristics and the demographic variables. And then we also want to see, most importantly, do these patterns at baseline, so upon intake, do they predict a response to treatment or speech language therapy? And in this case, we are reanalyzing data from a clinical trial that CSTAR conducted where it was TDCS applied in conjunction with speech language therapy. So they did that for, I believe, a month, and they were measured again 6 months later. Their performance was measured 6 months later. So what does our pipeline look like when we're trying to get at these questions? So again, we just have a T1 and a T2 as the scans that we are using for this study. So this is an example participant. we first have to enantiomorphically heal the brain. So what that means is we take the tissue from the healthy right hemisphere and fill in the stroke lesion with that tissue. And we do that because then it allows the ball-brain pipeline to correctly parcellate and estimate the brain ages of these individuals with stroke lesions. And the output of that is then a right hemisphere brain age gap map for each participant. So we can see here this participant had decelerated aging in kind of parietal lobe areas, and then accelerated brain aging in frontal and occipital areas. And then in addition to that, we also want to use information from the damaged left hemisphere. So for that, we do a very traditional classic approach. We have the left hemisphere lesion proportion map. So areas in yellow are areas that are highly damaged. And then as we go towards purple, those are areas that have less damage from the stroke lesion. And then we want to use a very similar approach to what we were using in the normative sample, and that's to use a dimension reduction approach to identify patterns of stroke lesions in the left hemisphere and patterns of brain aging in the right hemisphere. And what should result in biologically sensible patterns. So what do I mean by that? Well, for the stroke lesion, we know that stroke lesions are not randomly distributed throughout the brain. They follow arterial territories, right? So when we use this dimension reduction approach, we should find patterns of stroke lesions that align with what we know about the brain's arterial territories. And that's what we found. So 6 different patterns of commonly expressed stroke lesions emerged. And these patterns roughly aligned with arterial maps that we have, right? So these look biologically plausible. And I want to highlight that we also, in order to validate that this is not just from the sample that we were using, we did split half resampling. So we cut our participants in half, ran the factor analysis in one half, ran it in the other half, and saw if the patterns replicated in both halves. And we did that many, many times. And these patterns were stable across the split half resampling. And then we want to do the same thing for the right hemisphere. So these are the patterns that emerged using our dimension reduction approach. And these are patterns of brain aging in the right hemisphere of these stroke survivors. So factor 1 is kind of a medial prefrontal aging pattern. Factor 2 is really interesting because it looks like the homologs of the language cortex in the left hemisphere. Factor 3 was kind of a ventral pattern, but mostly cerebellum. And then factor 4 was this dorsal motor stream. And then what does this look like for our statistical models that we'll use? So these are our the predictors that we will put into our models for each participant. So we have the lesion scores, which represents how closely their lesion aligned with one of the commonly expressed patterns that we identified. And then we also, for the right hemisphere, have the brain age scores. So how closely their pattern of brain aging aligned with the gradients that we extracted. And so these are just individual participants shown as examples. So participant 1112 had a lesion score that was high in factor 3. And if we go back to the map, factor 3 is this pattern of basically a temporal damage in the left hemisphere. So we wanted to use these in a hierarchical linear regression. So our base model included a kind of global covariates that we would think would be related to aphasia severity at baseline. So lesion volume, chronological age, and sex. And then the next block of predictors are the scores for the different lesion factors. And the third block are the brain age factors. So we add these to the model to see how much variance we can explain in baseline aphasia severity. And And we can see here that adding the brain age factors to the model does explain more variance, and that is a significant change in the variance explained. And then in a subset of individuals with the Philadelphia naming task, we found similar results. And I want to highlight, that should be on brain age factor 1. So brain age factor 1, which if we recall is accelerated medial frontal aging, was related to performance in both the WAB-AQ and the Philadelphia naming task. So that factor 1 pattern seems to be very important for baseline aphasia severity. Okay, so we found that at baseline, and now we want to see if we can predict longitudinal treatment change. So in the clinical trial that was conducted. And we used a similar approach, except for the longitudinal participants, we only had, I think, 34 of them. We used the predictors that emerged as significant for the PNT in our first sample, and then we only used those 3 predictors in the longitudinal cohort. So this was, you can think of this as kind of a feature selection in an independent sample, and then we had the longitudinal people, which were completely separate. And these are the things that were related to naming in our baseline cohort. Now let's see if they are related to naming change in our treatment cohort. And what we found was that the model, including chronological age, lesion volume, and the scores for that brain age factor 1, did significantly predict naming change 6 months later. So it explained 23% of the variance in the naming change. And that was significant. And then the variance explained specifically by brain age factor 1 was also significant within this model. And this survived even when controlling for total right hemisphere gray matter volume, and also when controlling for baseline PNT score, which we know baseline performance can be an important predictor of longitudinal performance. But in this case, that did not negate the results that we found with this specific brain age pattern. And we can see here that this is an example responder in this cohort. So this participant had a large gain in naming, and they also did not have the medial prefrontal aging. So the take home is basically that participants who have this brain age pattern of accelerated medial prefrontal aging are less likely to respond to the treatment that was used in this clinical trial. So we found that the brain age gradients were associated with aphasia severity and treatment response. Again, I want to highlight that these are very simple inputs, so it is a scalable approach that can be used in larger cohorts or even to retrospectively analyze cohorts that only have T1s or T2s, for example. And some future steps could be using a brain age informed stimulation protocol. So perhaps mapping brain age patterns, see that they have a healthier parietal cortex, and then choosing that as a stimulation site for TMS or TDCS, for example. And then also looking at what are the modifiable factors that influence brain aging post-stroke. And just looking forward to the future, I'm working on a manuscript with my colleague Eleanor to make this very easy to use and automated pipeline for other researchers that want to apply this method to stroke cohorts. And thank you, everybody. Julius, Chris, Leo, Sarah, Ainsley, NIH, and of course, all of the participants and speech language pathologists.
All right, thank you so much, Nick. We can take some questions if there are any in the room. We have some online. Any questions from the room? I got a couple of questions while there was that little thought. The brain aging is a really, it seems like a very simple, and it sounds like a very clinically useful measure, whether it's the single number or regional brain aging. But can you talk a little bit about sort of what we know about these modifiable factors that may affect longitudinal brain aging? So, like if we have a stroke patient with aphasia, we see some pattern of regional brain aging. Is there any, I don't know, lifestyle treatment? Like, can you prescribe exercise or cessation of smoking that you know would improve or reduce their brain age gap in specific regions or overall?
Yeah. So that's a great question. And part of our ongoing research here is to see what is modifiable and what is not. And that can be tricky to disentangle. What I think we've been finding and a lot of other groups have been finding is that cardiovascular risk factors are very important for brain health. So, we're talking about BMI, cholesterol, blood sugar, blood pressure. So these are all things that are partially modifiable and are the things that we can kind of control when looking at brain health. So that's what I would say. I'm not sure about prescribing exercise or if we're at that point yet, but those are the factors that seem to be the most important.
Perfect, thank you. So we got a question in the chat from, and excuse my pronunciation, Hachamar, Benamino Hachamar, sorry. They say, I'm curious if there have been recent advances in studying brain aging patterns with functional MRI, especially in how effective connectivity between regions changes with age or brain age.
Yeah. So that's also a great question. Most of the traditional brain age models do use just structural MRI. James Cole and some other groups as well have tried to use functional neuroimaging as its own brain age. So how old does the activity in your brain look like at a very basic level? And I think that those have proven to be less reliable. That might be a bad word. They're less accurate at predicting chronological age because there is more variability in those functional patterns across individuals. So they're a little bit trickier to use in this way, but there are efforts going towards that, and also using multimodal brain age measures. So combining fMRI, DTI, and then just anatomical T1s to try to make a whole picture of brain health. In my experience, and I think reflected in what the field has been doing, I think the structural T1 measures have been the most fruitful up until this point. So, but yeah, definitely a room for expansion.
Perfect, thank you. Any questions here? Yeah.
I know you said that there were like 6 different patterns that were occurring, like the way that brains age. I was just wondering if there were any similarities between each of those groups for like geographic region, socioeconomic. Like, I just wasn't sure if there was something that like tied each of those groups together.
Yeah. So we'll go back to the patterns. The dimension reduction approach that we used forced the patterns to be as different from each other as possible. So it forces orthogonality in the data. So if you look at an individual participant, they may have a high score in multiple patterns. So they might express multiple patterns, but at a group level, the patterns themselves are not correlated with each other. And I think that that's a strength of this approach, but also could be a limitation since we are trying to force the patterns to be as different from each other as possible. There are other approaches you could use that would find patterns that are more like each other. But in regards to SES, these participants have a fairly low range of SES scores. They're actually all pretty similar. These are people from the aging brain cohort mostly. So definitely room for expansion. I think Natalie Busby has published a paper on socioeconomic status and brain aging, but she did not use these patterns is global brain age. So definitely an area that we're looking into.
Maybe just to piggyback off of that question and looking at these maps, you mentioned that there was an association between one map and visual acuity, right? So relating differences in regional brain aging to behavioral outcomes. Do you know, and I know this is an evolving literature. Do you know how much of that is, or what direction the causality goes? Is it because there is advanced brain aging in the occipital cortex that the visual acuity is bad, or is the regional brain aging in the occipital cortex advanced because they have poor visual acuity?
Yeah. Yeah, that's a great question. And I don't want to speculate because I don't know. Again, we want to use this in a longitudinal data set in order to see if, the brain aging does come before the declines in cognition or whatever. But we have not done that yet. So I think that would be very powerful to know the answer to that question.
Thank you.
Sorry, I missed the first half of the talk. I'm curious, to what extent, the variance in brain age, predicted brain age is associated with the actual biological age, how much variance between them.
Yeah, it depends on the model that you use. This ball brain model, I believe that the R squared, they train their model on ages 5 to 85, and the R squared between predicted brain age and chronological age was, I think it was like 0.65 or something like that. Their specific model is more accurate in younger adults. And then as people get older, the model becomes less accurate. And I'm putting accurate in quotes because a perfect brain age model would be a useless brain age model. What we're interested in is actually the gap when it is, not predicting well. Is that difference biologically meaningful, right? But yeah, I think a lot of the models have very good performance. So it is accurately predicting chronological age.
Maybe one more question. So that relates to the stroke work you've been doing. And again, I know this is a young literature, but can you speak a little bit about what we know about the association between lesion, both location and the size of the lesion, and sort of brain aging patterns in the contralateral hemisphere? And keeping in mind that we know from the literature that people who have a stroke at an older age, they tend to correlate with those who have a higher brain age to begin with. So again, how much is related to the stroke? How much does that advance their brain age?
Yeah. So I didn't show this in this talk, but we did conduct another subset of analyses looking at those questions. So how does lesion volume, the location of the stroke lesion, how do those relate to the expression of these brain age patterns? And we did find relationships between some of them. So it did seem that people with a certain pattern of stroke lesion did tend to express different patterns of brain aging. So there are relationships there. But again, a little hesitant to say that the stroke lesion is causing a certain pattern of brain aging, even though they are significantly associated, just because we have not yet looked into the longitudinal data kind of showing if, for example, if somebody with a stroke lesion in the temporal lobe, if we follow that person over, months and months and months, do they express more and more and more brain aging in a certain region? We haven't looked at that yet, but.
Okay. Thank you. All right. If we have no more questions, then.
May I
we'll
ask
give
a
it
quick
up for Nick.
question?
Oh, thank you. One
Hi,
more.
sorry.
Go ahead, Karen.
Oh, yeah. So Nick, thanks for your talk. I really enjoyed it. When you were talking about exploratory factor analysis, you said that you did split half or cross data set for the replication? And then did you use a certain rotation or factor number criteria?
So factor number criteria was set on the eigenvalue being greater than 1. Rotation was very max rotation because we wanted maximally orthogonal patterns that we could put into a linear regression. So I have tried different types of dimension reduction, so PCA and different types of rotations. And these patterns generally emerge regardless of what type of dimension reduction approach you are using.
Okay, yeah, that's been tricky for me, and I'm not even doing I'm only doing behavioral type measures. So yeah, that's very helpful. Thank you.
All right. Thank you so much, Nick. All right. Thank you again for a great talk. This brings us to the end of today's talk. Thank you so much once more to Dr. Riccardi for a great talk, and thanks to our online and in-person audience for asking questions and for watching. We look forward to seeing you again in 2 weeks' time for another C-Star lecture.